Okay, this is a walkthrough of how to model out mitosis and meiosis. And we need to know a few things about the terminology with the pieces of our model that we're going to be working with. So, hand down here. And first thing is our string of pop beads here. This is the middle magnetic part will be the centromere of our chromatids, or each of these would be a chromosome. Like separately, this is a chromosome. This is a duplicate chromosome as well. And when they're together like this, this is still one chromosome, but they're known as sister chromatids because they are actually held together at their centromere, right? So we have sister chromatids right here. Okay, and so we're going to do a simple model with a cell that is just two chromosomes in its diploid number. So 2n will equal 2. It will have a pair of homologous chromosomes, one from the father and one from the mother. So let's go ahead and get started. Okay, so first we're going to look at mitosis. Okay, so mitosis is going to be where we go through DNA replication during S phase, and then there'll be one cell division. All right, so we're going to work through mitosis. All right, the first phase, we're going to look at going through S phase, which is prior to actual M phase and getting into mitosis. And so we're going to have a cell. All right, so here's our cell. And inside the cell we have a nucleus, okay, and next to that nucleus we're going to have a pair of centrioles, which makes up a centrosome. This is found in animal cells. And then we'll also have our couple homologous chromosomes. All right, now these guys won't be hanging out like locked in together, but during interphase they will be found as chromatin, which is simply like a string-like expanded form of our DNA information. All right, so what will happen during S phase is these will actually get replicated. All right, and so we build a copy of one of our chromosomes, and we then build another copy of a different chromosome, and these are now they now have their sister chromatids attached at the centromere. Right. Also, the centrioles will get duplicated, and so they'll be hanging out just outside the nucleus. So we're getting right up to the point of going into mitosis. All right, the first stage in mitosis is going to be prophase, and in prophase, what happens? The centrioles start to migrate to other sides of the cell. The nucleus, the nuclear membrane, fades away, kind of breaks apart, and these chromosomes now are the condensed form of chromatin. So what used to be all like squiggly spaghetti-like DNA loosely packed, now it's tightly coiled and packed and wrapped up in the traditional X shape that we see once we have a replicated chromosome. Right? So we have sister chromatids now, and if we looked at it under a microscope, we could see all this. Okay, also from these centrioles, we will have spindle fibers starting to connect to the sister chromatids here at the centromere, and this is to help get them lined up into the next phase. So as that happens, the centrioles will make their way to the sides of the cell, and they'll start to put these chromosomes in line at the metaphase plate. So this metaphase plate is going to be roughly on the center of the cell between the two centrioles, and there will be spindle fibers that will attach to either side of the centromere that's shared by these sister chromatids. Right, so we have our spindle fibers in place, and now we are in metaphase. Okay, notice that we are lined up with the total number of chromosomes. We have two chromosomes in this cell. It's a diploid cell with two chromosomes, 2n equals 2, and now we are ready to move into anaphase. And anaphase is going to be where these spindle fibers, the kinetochore in the centromere, will motor these sister chromatids apart. So they break apart and they start heading towards opposite sides of the cell. Right, notice that we have the telomeres, the long telomeres, will kind of drag behind the direction that these now daughter chromosomes are headed. Right, they were sister chromatids when together, but now that they're apart, they are their own copy of the genome. Right, now they are independent of one another. As they make their way to the far sides of the cell near the centrosomes, or the pair of centrioles at either end of the cell, we now get into telophase. And in telophase, these guys are not going to combine or link up or click together, right? They're going to stay their own separate chromosomes. But what does happen is we see the reformation of a nuclear membrane. All right, so I'm going to get these guys out of the way for a second. I'm going to draw a new nuclear membrane, and I'll try to put these guys in here without making them snap together. All right, then the centriole will stay right outside that nuclear membrane. Same thing will happen over here. So we have a new nuclear membrane that is formed, and our newly separated chromosomes are present. And these guys will start to uncoil and go back into the chromatin form, and also this centriole set will be just outside the nucleus. Right. We'll also notice that our cell membrane now has kind of elongated along the axis that the centrioles were at, and it actually starts to fold in on itself. I don't know if you can see that very well, but we'll make a little misshapen peanut almost. And this is where the cleavage furrow is going to start to form. Right, so this will be where we start to see the cells start to pinch their membranes together, and this is the end of mitosis. Right, to finish the job up, we will then move into cytokinesis, and that will be where we actually then separate these two into now their own daughter cells with a complete set of the diploid number of chromosomes, 1, 2, and 1 and 2. Right, and so that's mitosis. We start with a diploid cell, and the DNA is replicated just prior to mitosis, and we go through one cell division, which results in a diploid number in our daughter cells. And these cells would be duplicates or clones of the previous cell. Okay, so you may have just watched mitosis, but now we're going to go into meiosis. All right, so in meiosis, this is where we're going to do replication once during S phase in interphase of the cell, and then we're going to go through two cell divisions. So we're basically going to go through our PMAT process times 2. Right? So it's PMAT times 2. All right, so let's go ahead and get our cell set up. All right, first we'll go ahead and get our cell membrane drawn. Okay, so here's our wonderful cell membrane. And inside the cell membrane, obviously, we're going to have a nucleus. And just outside this nucleus, we will have a set of centrioles known as the centrosome, and this is found in animal cells. And we will also have, in our cell's case, we're just going to start with a very simple 2n equals 2. And these are homologous chromosomes, right? So they have the same similar genetic information, like maybe the gene for hair color, or dad's webbed toes and mom's not webbed toes, right? So these are homologous chromosomes, one from the paternal donor, one from the maternal donor. Okay, so in S phase, we're going to make a copy of these guys, just like we see happen in interphase of any cell, moving through S phase. So we'll make a replicate of one of our chromosomes here, and we'll make a replicate of the other. And then also during this stage, just prior to meiosis, the centrioles will replicate as well. All right, so very similar in the setup that we see prior to going into mitosis. We have gone through replication of DNA, so DNA synthesis has occurred, and we've also made a replication of our centrioles. Okay, so now this is going to be where things play out a little different when we're looking at meiosis. So we're going to go into prophase number 1. All right, so in prophase 1, the nuclear membrane is going to break down. All right, so we go ahead and we get rid of that. And the centrioles are going to start to migrate to opposite sides of the cell. So we'll go ahead and get that set up. Spindle fibers will start to spawn out from these, sometimes connecting to the other centriole. All right, so we have some spindle fibers moving along here. And what's different is that in prophase 1, we will find that these homologous chromosomes will actually find each other and meet up, like so. They form what is called a tetrad because you have 1, 2, 3, 4 chromatids in total, 2 pairs of sister chromatids, and they're very, very close together with their centromeres kind of interlocked. And what will happen is we're going to get some crossing over that's going to happen, right? So these guys will kind of get entangled with each other, and we're going to simulate crossing over by popping off 3 of our beads here and 3 of our beads off of this guy, and they're going to actually swap this genetic information. Now, this can happen more than once. This can happen in several locations, but to keep this model simple, we're just going to show one crossing over event right here. All right, so now, this is what kind of fills out the process in prophase 1. Now we're going to go into metaphase 1. In metaphase 1, these centrioles have made their way to opposite sides of our cell. Okay, we'll try to keep that on camera. Okay, and then this is where we have another difference when it comes to the meiosis process. All right, so in metaphase 1, the entire tetrad is brought into place and positioned at the midline of the cell by these spindle fibers. All right, so we just had a diploid number of 2n equals 2, so we have just one tetrad to worry about. But in like a human cell, there would be 23 of these tetrads lined up because we have a total of 46 chromosomes, 23 pairs of homologous chromosomes. All right, so now this is now metaphase 1 of meiosis, and the crossing over has completed. And what we're going to see then next is anaphase 1, where the spindle fibers will pull, and we're going to separate the tetrad. So in anaphase 1, we're actually pulling the entire sister chromatid set in 2 different directions, right? So one set will go to this direction, the other set will go the other direction. All right, as they near the centrioles, we reach telophase 1, and just like before, we're going to have these centrioles kind of move a little bit. The nuclear membrane will reform, so we'll put a new nuclear membrane around these chromosomes. All right, and we're going to see, just like in telophase and mitosis, we're going to get a kind of peanut shape going on with our cell membrane as it starts to pinch in, and we get a cleavage furrow between the new eventually forming daughter cells. Right. So this telophase process completes, and now we go into cytokinesis. And in cytokinesis, we will go ahead and split these guys into 2 separate cells. All right, so now we have 2 entirely independent now daughter cells. All right, notice at this point, each daughter cell contains 1 chromosome. Right, these are sister chromatids. Right, they are no longer identical because we've had crossing over occur. Right, that adds the genetic variation, but we only have 1 chromosome in each of these daughter cells. So we are now at the haploid number, which is 1 step closer to being gametes. All right, so now what we need to do is, through a brief rest, a very brief rest between the end of cytokinesis and into prophase 2 of meiosis, the centrioles will actually go through another replication. So I'll grab a few more. Right, so we have another set of centrioles to use. And now what we're going to do is work with both of these daughter cells and what's going to happen inside of them. Right, now to do that, I'm going to I'm going to go ahead and make our daughter cells a little bit larger, so bear with me. Okay, so now we're ready to go into prophase 2. So in prophase 2, these chromosomes are going to recondense, make sure that they're coiled back up into this typical chromosomal format, right? And we have our sister chromatids that are not quite identical anymore. And the centrioles are going to start to make their way to opposite sides of the cell. And at the same time, this nuclear membrane is going to break down. So we'll get rid of the nuclear membrane. All right, and spindle fibers are starting to spawn out and connect to the chromosomes that we have. And this kind of is the end of prophase 2. Now as we make our way to metaphase 2, the centrioles make their way to opposite sides of these respective daughter cells. The chromosome is now lined up at the metaphase plate, and this one will do the same. Okay, and don't get misled that these centrioles can line up at opposite sides at any opposite ends of the cell. I just have them lined up in this direction. All right, and so we now have our haploid set of chromosomes, in this case just one chromosome, in metaphase 2 lined up on the metaphase plate. And we have spindle fibers that are attaching to the centromere here of these chromosomes. And exactly what you think will happen next. In anaphase 2, we're actually going to pull and separate the sister chromatids. So this is going to happen in both of these daughter cells. All right, as we make our way to each centriole, we now reach telophase or telophase. And just as before, as we go into telophase, we're going to have the formation of a new nucleus. So we'll get a new nucleus around this guy. I'm going to try to leave ourselves some space. All right, and then we'll do the same thing over here. A new nuclear membrane. A new nuclear membrane. Okay, so now we have, we're at the end of telophase, and finally, what you expect to happen is we're going to go through cytokinesis so that each of these nuclei have their own cell membrane. So I'll do a little bit of cleaning up and we'll do some video magic. Okay, so now we have 4 daughter cells, right? And they each have their own nuclei. And in each nuclei, there's one chromosome, and one chromosome, and one chromosome, and one chromosome. So we have now produced the 4 haploid gamete cells originating from one diploid cell going through meiosis. Right? So the final process or step was to go through cytokinesis one more time here to split these separated sister chromatids, now daughter chromosomes, into their own respectful cells. Right? And so that is the end of meiosis. And hopefully this helps you get prepared and ready for that assessment where you want to show me how all of this plays out, whether it's mitosis or in this case, meiosis. So hope this helps you out. If you have any questions, be sure to ask in class.